Iron is an essential metal ion for growth, making it in high demand for many organisms. The increase of oxygen in the Earth's atmosphere led to a lot of previously soluble iron 2+ being locked away in insoluble iron oxides, beginning the biological war for soluble iron. One evolutionary result of this is the production and secretion of siderophores by certain types of bacteria. Siderophores are compounds that bind iron 3+ with high affinity, with examples shown here with the coordinating groups in blue. These are examples of tetradentate siderophores, meaning they bind through 4 groups. These siderophores are secreted outside the bacterial cells, and once having bound iron, they can be transported into the periplasm of the cell through receptors on the surface. Within the periplasm, the iron siderophore complex can then bind to a periplasmic binding protein, which is then taken up into the cytoplasm of the cell through a transport protein in the membrane, where the iron can then be released. Researchers at York are interested in understanding this mechanism and exploring the interactions between periplasmic binding proteins and iron siderophore complexes and their potential applications. Small crystals of the siderophore bound to the protein were grown, and a map of the structure was able to be determined using X-ray crystallography. The key interactions observed are the side chains of histidine and tyrosine residues involved in the coordination of the metal ion in an octahedral geometry, as well as electrostatic interactions between the positively charged arginine side chains and the negatively charged siderophore complex. The arginine side chains also act as hydrogen bond donors to several of the coordinating oxygens of the siderophore, with the hydrogen bonds being shown as dashed black lines. The crystal structures of other periplasmic binding proteins also contain histidine and tyrosine residues in similar places, suggesting their role in binding is crucial, and hence why they are conserved across each of these structures. Researchers at York have been able to use the knowledge of these interactions in different applications, such as in artificial metalloenzyme design. The high binding affinity of siderophores to periplasmic binding proteins has been used to anchor organometallic catalysts to protective protein scaffolds. Proximity to the protein allows for enantioselective catalysis, where one isomer of product is formed preferentially to another, which is not possible when the catalyst is used alone. The bacterial uptake mechanism of iron siderophore complexes also presents another route for drug delivery. Attachment of antimicrobial drugs to siderophores allows for a Trojan horse strategy as a method of combating antimicrobial resistance, with several siderophore drug conjugates entering clinical trials. These applications highlight this area of bioinorganic chemistry as promising for future research and development.